Hello friends, so today we are presenting a topic that is about the resistance of the body to infection, that is immunity and allergy. Here is the outline of immunity and allergy. 1. Innate immunity and acquired immunity. 2. Specific attributes of the B-lymphocyte system, humoral immunity, and antibodies. 3. Complement system activated by antigen, antibody reaction. 4. Special attributes of the T-lymphocyte system, activated T cells, and cell-mediated immunity. 5. Types of T cells, and their different functions. 6. Immunologic tolerance. 7. Allergy and hypersensitivity. Let's start. First, we will discuss innate immunity and acquired immunity. The immune system is typically divided into two categories: innate immunity and adaptive or acquired immunity, although these distinctions are not mutually exclusive. Innate immunity refers to nonspecific defense mechanisms that come into play immediately or within hours of an antigen's appearance in the body. These mechanisms include physical barriers, such as skin, chemicals in the blood, and immune system cells that attack foreign cells in the body. The innate immune response is activated by chemical properties of the antigen. Adaptive immunity or acquired immunity refers to antigen-specific immune response. The adaptive immune response is more complex than the innate. The antigen first must be processed and recognized. Once an antigen has been recognized, the adaptive immune system creates an army of immune cells, specifically designed to attack that antigen. Adaptive immunity also includes a memory that makes future responses against a specific antigen more efficient. Here is a brief difference between innate and acquired immunity cells for its better understanding. Let's learn the differences between these two in detail. 1. Innate immunity is something already present in the body, while adaptive immunity is created in response to exposure to a foreign substance. 2. Innate immunity is nonspecific. On the other hand, adaptive immunity is specific. 3. Innate immunity fights any foreign invader, whereas adaptive immunity fights only specific infection. 4. Innate immunity is rapid in response, in contrast to adaptive immunity, which is slow in response, that is, it takes 1 to 2 weeks to mount a specific response. 5. Innate immunity has limited and lower potency, while acquired immunity has high potency. 6. Once activated against a specific type of antigen, the innate immunity remains throughout the life. On the other hand, the span of developed immunity can be lifelong or short. 7. Innate type of immunity is generally inherited from parents and passed to offspring, whereas adaptive immunity is not passed from the parents to offspring, hence it cannot be inherited. 8. Innate immunity cannot react with equal potency upon repeated exposure to the same pathogen. On the contrary, adaptive system can remember the specific pathogens which have encountered before. 9. Innate immunity is present at birth, while adaptive immunity develops during a person's lifetime and can be short-lived. 10. In innate immunity, there is no allergic reaction. On the other hand, adaptive immunity shows immediate and delay hypersensitivity. 11. Innate immunity is used for microbes, while adaptive immunity is used against microbes and non-microbial substances called antigens. 12. Innate immunity has no memory. On the contrary, adaptive immunity has long-term memory. 13. Innate immunity has limited diversity, while adaptive immunity has high diversity. 14. Innate immunity has faster response, while adaptive immunity has slower response. 15. Alternative and lectin pathways are the prominent pathways required for innate immunity. On the other hand, classical pathway is the dominant complement pathway required for adaptive immunity. 16. Anatomic and physiological barriers provide the crucial first line of defense against pathogens. These barriers include skin, mucous membranes, temperature, pH, chemicals, etc. On the contrary, the anatomic and physiological barriers of adaptive immunity include lymph nodes, spleen, mucosal-associated lymphoid tissue. 17. The innate immune system is composed of physical and chemical barriers, phagocytic leukocytes, dendritic cells, natural killer cells, and plasma proteins. While adaptive immune system is composed of B cells and T cells. 18. Innate immunity is evolutionary, older, and is found in both vertebrates and invertebrates. On the other hand, adaptive immunity system has been developed recently and is found only in the vertebrates. 19. Example of innate immunity include white blood cells fighting bacteria, causing redness and swelling when a person has a cut. While example of adaptive immunity include chickenpox vaccination so that we don't get chickenpox, because adaptive immunity system has remembered the foreign body. Next we will discuss specific attributes of the B-lymphocyte system, humoral immunity, and antibodies. First we will learn how antibodies are formed by the plasma cells. Before exposure to a specific antigen, the clones of B-lymphocytes remain dormant in the lymphoid tissue. On entry of a foreign antigen, macrophages in the lymphoid tissue phagocytize the antigen and then present it to adjacent B-lymphocytes. In addition, the antigen is presented to T cells at the same time, and activated helper T cells are formed. These helper cells also contribute to extreme activation of the B-lymphocytes, as we discuss more fully later. Those B-lymphocytes, specific for the antigen, immediately enlarge and take on the appearance of lymphoblasts. Some of the lymphoblasts further differentiate to form plasmablasts, which are precursors of plasma cells. In the plasmablasts, the cytoplasm expands, and the rough endoplasmic reticulum vastly proliferates. The plasmablasts then begin to divide, at a rate of about once every 10 hours, for about 9 divisions, giving in 4 days, a total population of about 500 cells for each original plasmablast. The mature plasma cell then produces gamma globulin antibodies at an extremely rapid rate, about 2,000 molecules per second, for each plasma cell. In turn, the antibodies are secreted into the lymph and carried to the circulating blood. This process continues for several days or weeks until finally exhaustion and death of the plasma cells occur. What is the difference between primary and secondary response? Let's learn. A few of the lymphoblasts, formed by activation of a clone of B lymphocytes, do not go on to form plasma cells, but instead form moderate numbers of new B lymphocytes, similar to those of the original clone. In other words, the B cell population of the specifically activated clone becomes greatly enhanced, and the new B lymphocytes are added to the original lymphocytes of the same clone. They also circulate throughout the body to populate all the lymphoid tissue. Immunologically, however, they remain dormant until activated, once again, by a new quantity of the same antigen. These lymphocytes are called memory cells. Subsequent exposure to the same antigen will cause a much more rapid and much more potent antibody response this second time around, because there are many more memory cells than there were original B-Lymphocytes of the specific clone. How complement system is activated by antigen-antibody reaction. Let's discuss. There are 3 ways to activate the complement system, involving different molecules initially, but converging to produce the same effector molecules. Each involves activation of enzymes that cleave their substrates to form a cascade, so that the complement response is amplified. These include 1. The classical pathway. 2. The Mannose-Binding lectin pathway, and 3. The alternative pathway. All 3 pathways produce C3 convertase, an enzyme, which triggers further effects downstream. Now, we will discuss the effects of C3 convertase in detail. First one is the classical pathway. The classical pathway is activated when a complement protein called C1q binds, either directly to a pathogen or onto an antigen-antibody complex. This will then trigger cleavage of the subsequent complement proteins in the cascade, resulting in production of C3 convertase and its downstream effects. Its involvement in antigen-antibody complexes means it has a role in the adaptive immune response as well as the innate. Next is the Mannose-Binding lectin (MBL) pathway. Mannose-Binding lectin or MBL is a protein produced in the liver. Its role is to detect carbohydrates containing mannose on the surface of pathogens, activating a protease called MASP. MASP is responsible for cleaving complement components, which activates a similar cascade to the classical pathway, eventually producing C3 convertase. Third one is the alternative pathway. The alternative pathway is usually activated by bacterial endotoxin, a lipopolysaccharide present on the outer membrane of gram-negative bacteria. This results in spontaneous hydrolysis of C3 into small amounts of factor C3b, which combines with other factors to produce C3 convertase. Whichever way C3 is activated, it will then activate C5, which in turn activates C6, C7, C8, and C9 in a cascade. As such, even a small signal can lead to the rapid activation of many thousands of complement molecules. This is important in the immune response, as pathogens are also able to replicate very quickly within the body. Once activated, the complement system has several effects, including opsonization, lysis of pathogens, chemotaxis, and inflammation. We will discuss them one by one. What is opsonization? Let's learn. C3 convertase is a product of all the pathways triggering the complement cascade and is responsible for converting factor C3 into C3a and C3b. C3b binds to antigens on the pathogen, which stimulates neutrophils and macrophages to phagocytose pathogens. This is called opsonization. Upcoming is lysis of pathogens. Lysis of pathogens is facilitated by the formation of the membrane attack complex, MAC. C3 convertase is vital to the production of the MAC because it generates C3a and C3b. C3b combines with other factors to produce C5 convertase, an enzyme which converts factor C5 to C5a and C5b. C5b combines with several factors to produce the MAC. The MAC ruptures the bacterial cell membrane, allowing fluid to enter the bacteria and causing cell lysis. However, because they possess a cell wall, gram-positive bacteria and fungi do not swell, and hence cannot be lysed by the complement system. What is chemotaxis? The production of C5a by C5 convertase attracts neutrophils and macrophages to the site of infection and causes extravasation of leukocytes from capillaries to tissues. C3a is another complement component that acts as a chemotaxin. Which complement components are responsible for causing inflammation?
C3a, C4a, and C5a are the complement components responsible for causing inflammation.
They bind to mast cells and basophils to cause degranulation. The histamine and serotonin released increase vascular permeability.
C3a, C4a, and C5a also promote synthesis of pro-inflammatory cytokines.
What is activated T cells and cell-mediated immunity? Let's learn. The immune system distinguishes two groups of foreign substances. One group consists of antigens that are freely circulating in the body. These include molecules, viruses, and foreign cells. A second group consists of self cells that display aberrant MHC proteins. Aberrant MHC proteins can originate from antigens that have been engulfed and broken down that are exogenous antigens, or from virus-infected and tumor cells that are actively synthesizing foreign proteins that are endogenous antigens. Cell-mediated immunity is an immune response that does not involve antibodies, but rather involves the activation of phagocytes, antigen-specific cytotoxic T-lymphocytes, and the release of various cytokines in response to an antigen. The principal role of cell-mediated immunity is to detect and eliminate cells that harbor intracellular pathogens. The antigen-specific cells contribute to the cell-mediated immunity, include CD8+ cytotoxic T-lymphocytes or CTLs, and cytokine-secreting CD4+ Th or T helper cells that mediate delayed type hypersensitivity. Effector T cells, generated in response to antigen, are responsible for cell-mediated immunity. Both activated T helper cells and cytotoxic T lymphocytes or CTLs play effector cells in cell-mediated immune reactions. Cytokines, secreted by T helper cells, can activate phagocytic cells and enabling them to phagocytose and kill microorganisms more effectively. The antigen nonspecific cells that contribute to the cell-mediated immunity include NK cells, macrophages, neutrophils, and eosinophils. T cells, NK cells, and macrophages are the most important sources of the cytokines that organize and support cell-mediated immunity. Cell-mediated immunity can recognize and eliminate tumor cells that have undergone genetic modifications so that they express antigens not typical of normal cells. Though humoral and cell-mediated immunity have many distinctive features, they are not completely independent. Macrophages, NK cells, neutrophils, and eosinophils can use antibodies as receptors for killing target cells. Activation of complement system by antigen-antibody complexes gives rise to chemotactic peptides, which help in assembling the cell types required for a cell-mediated response. Next, we will discuss types of T cells and their different functions. What are T cells? Let's discuss. T cells, or T lymphocytes, are a major part of the immune system. They are cells specifically designed to fight infections they have not yet encountered. As part of any research, there may be a time when one needs T cells for isolation. They mature in the thymus until released into the bloodstream, as naive T cells. Naive T cells are unassigned T lymphocytes that search for an antigen-presenting cell or APC. How are T cells activated? When a T cell encounters a recognizable APC, the naive cell receives a signal to mature.
There are three types of signals: TCR, BCR, and cytokine signals.
If a cell receives all three signals, it will mature into an effector cell. If a cell only receives one of the signals, TCR or BCR, the cell will become useless. What are effector cells?
Depending on the APC, a naive cell comes across it, can become an effector T cell.
Effector T cells have relatively short lifespans and carry out the functions of an immune response. They can be cytotoxic, helper, and regulatory T cells. What are cytotoxic T cells? Cytotoxic T cells, also known as CD8+ cells, have the primary job to kill toxic or target cells. Upon recognition, their purpose becomes the removal of virally infected cells, bacteria, and tumor fragments, such as cancer cells, through a process called apoptosis. Apoptosis occurs when a cell's internal organelles are destroyed, causing it to die from the inside out. What are helper T cells? T helper cells, or CD4+ cells, are similar to cytotoxic cells, but have a wider range of functions. These cells are extremely important to cell immunity because they are required for most adaptive immune responses. T helper cells become activated when presented with antigens and have the ability to differentiate into cell subtypes. When activated, helper T cells multiply and secrete cytokines that summon macrophages and cytotoxic T cells to the infected site. What are regulatory T cells? The final type of effector cell is the regulatory T cell. Regulatory T cells are tasked with stopping an autoimmune response once the threat has been eliminated. After helper T cells and cytotoxic T cells bind to a pathogen and work together to rid the body of it, they no longer serve a purpose. Regulatory T cells stop them from taking up space or accidentally attacking healthy cells until they succumb to apoptosis. What are memory T cells? While these three types of effector cells handle the main immune response, they are not the only variations of T cells. Some T lymphocytes are present even after removing a pathogen. These long-living lymphocytes are memory T cells and are highly capable of responding to antigens upon reintroduction. These cells are formed after an infection and are extremely important because they have the ability to expand into large numbers of effector T cells upon exposure to familiar antigens. Memory cells are unique because they remember pathogens and infectious cells faster than others, allowing them to fight off bacteria and viruses quickly. Memory T cells are the reason vaccines can create immunities in the body. How the study of these different types of T cells can be applied. Each of these T cell variations can help us to learn about diseases and our body's responses to them. Naive T cells teach about adaptability, effector cells give ideas for methods of treatment, memory T cells assist in creating vaccines and medicine. T cells are an essential part of the medical research field and could pave the way to many breakthroughs in the near future. Upcoming is Immunologic Tolerance. Immunological tolerance describes a diverse range of host processes that prevent potentially harmful immune responses within that host. This is achieved by avoidance of adaptive immunity, such as the forms of immune privilege, seen in maternal acceptance of the fetus, in cancer, and in diverse body tissues, or through fine control of adaptive immunity, in lymphocytes, throughout their lifetime. An understanding of mechanisms underlying tolerance promises opportunities to therapeutically reprogram the immune system, to treat autoimmune diseases and allergies, to ensure improved acceptance of transplants, and to provide better vaccines against microbes and cancers. Immunologic tolerance is an active, but carefully regulated, response of lymphocytes to self-antigens. Normal individuals are tolerant to their own antigens or self-antigens. Even though many self-antigens have free access to lymphocytes, lymphocytes do not normally mount an immune response against self-antigens. This self-tolerance is maintained by several mechanisms that prevent the maturation and activation of potentially self-reactive lymphocytes. Immunologic tolerance occurs in two forms, central and peripheral. Let's compare central and peripheral immunologic tolerance with respect to B-cell and T-cell participation and how, when, and where these occur. Central tolerance refers to the tolerance established by deleting autoreactive lymphocyte clones before they develop into fully immunocompetent cells, while peripheral tolerance develops after T and B cells mature and enter the peripheral tissues and lymph nodes. In central tolerance, lymphocytes that do not receive survival signals undergo apoptosis. On the other hand, peripheral tolerance can occur through one of three mechanisms. A. Induction of energy. It is a state of inactivation, in which the lymphocytes remain alive, but are unable to respond to antigen. B. Apoptotic death or deletion of mature self-antigen-specific lymphocytes. C. Suppression of lymphocyte activation and functions by regulatory T cells or by other immunosuppressive effectors. Lymphocyte development and education is most active in fetal development, but continues throughout life, as immature lymphocytes are generated, slowing as the thymus degenerates and the bone marrow shrinks in adult life. Peripheral tolerance continues throughout life. Central tolerance occurs during lymphocyte development, in the thymus and bone marrow for T and B lymphocytes, respectively. While peripheral tolerance occurs in whole organism, except thymus and bone marrow. Last topic that we will discuss is allergy and hypersensitivity. Allergy or hypersensitivity occur when the body's immune system reacts in an exaggerated or inappropriate way to substances in the environment that would not normally cause inflammation and disease. What causes allergy and hypersensitivity? Allergy and hypersensitivity are caused by abnormalities in the regulation of the immune system. In some cases, the immune system produces a kind of immunoglobulin or antibody called immunoglobulin E or IgE. This is the commonest kind of allergy. When IgE is not involved, these kinds of reactions are sometimes referred to as hypersensitivities. Unfortunately, the terms allergy and hypersensitivity are commonly used interchangeably, which is confusing. When the mechanism involves the production of IgE, its targets are against substances known as allergens, which can be breathed in. This can include pollens, house dust mites, molds or skin, hair, saliva from cats, dogs, rodents, or horses. Be eaten. This includes things such as milk, eggs, fish, shellfish, nuts, cereals, or fruit. Come into contact with the skin, for example, latex and gloves. be injected into the body. This can include drugs, such as antibiotics and anesthetic drugs or insect venom. Allergic reactions can be relatively mild, such as a hay fever reaction to pollen, or they can be strong and cause a life-threatening reaction, called anaphylaxis. What are the signs of an allergic reaction? These include sneezing, a runny nose, and runny eyes. This is caused by inflammation in the nose, referred to as rhinitis, or when it affects the eyes, it is called conjunctivitis. Trouble in breathing with a cough, wheezing, and breathlessness, such as in asthma. This is caused by inflammation of the airways. Skin reactions such as eczema or hives, a raised, red itchy rash, also known as urticaria. Swelling of the deeper layers of the skin, caused by a buildup of fluid. This is known as angioedema. Sensitivity to specific foods, leading to stomach pains and vomiting. One particular type of hypersensitivity, that can occur in primary immunodeficiency, or PID patients, is a reaction to blood products. This can be significant for small numbers of patients treated with immunoglobulin, particularly intravenous immunoglobulin products. Which PID conditions are particularly associated with allergy? IgA deficiency. This condition has an association with both allergy and autoimmune disease. Omenn syndrome, hyper-IgE syndrome, IPEX syndrome. In these last three conditions, the most common sign of allergy tends to be severe eczema. Granulomatous disease is a form of non-IgE hypersensitivity inflammation that is also important in some kinds of PID. Here we reach the finish line. For more informative videos, please subscribe our channel and like our Facebook page. See you.